Hello, we're going to continue our conversation about muscle contraction. So the next step in muscle contraction is called excitation-contraction coupling. And this is a sequence of events where that transmission of the action potential down the sarcolemma is going to cause those myofilaments, actin and myosin, to start to bind together and start to slide toward each other. It's called the sliding filament theory. So we have to get that action potential from the sarcolemma down into the muscle fiber, and we want to cause a release of calcium. So this release of calcium is our goal because you have to have calcium for the actual contraction, the actual actin and myosin to bind in order for those muscle fibers and sarcomeres to contract. So just to kind of briefly remind you of where we are, we had an action potential come down the axon to the axon terminal. That caused a voltage change, which opened up calcium channels. Those calcium channels led to the release of acetylcholine. Acetylcholine then crossed the synaptic cleft to receptors where it bound. And remember, these were ligand-gated receptors, which allowed sodium to enter into the cell and potassium to leave the cell, which generated a small depolarization. That depolarization on the membrane led to an action potential that was able to spread across the sarcolemma, which is the plasma membrane of the muscle cell. So now we need that action potential to spread into the muscle cell itself to start this release of calcium, which will allow actin and myosin to bind. So the step we are currently talking about is the action potential, but I'm sorry, I've lost my voice, going down the sarcolemma and into the transverse tubules. So here are the 4 steps that we're going to cover in detail. We're going to cover the action potential traveling down the sarcolemma into the transverse tubule. We're going to cover the release of calcium from the sarcoplasmic reticulum, the binding of calcium to troponin, and then the actual beginning of contraction. So we're going to blow these pictures up and talk to you in a little more detail about these steps. So the first step, you have the action potential propagating down the sarcolemma into the T tubule. Now there's something really important about the anatomy of this particular part of the muscle cell. Remember, your sarcoplasmic reticulum is the endoplasmic reticulum of the muscle cell, and it stores calcium. The sarcoplasmic reticulum makes these things called terminal cisternae, and they're kind of wide spots on the sarcoplasmic reticulum that abut the T tubule. So where you have a sarcoplasmic reticulum and a T tubule and then another sarcoplasmic reticulum, these 2 terminal cisternae, we call that a triad. Spell that, triad, so 3 things. And so because of this anatomy, this T tubule and the sarcoplasmic reticulum are really close to each other. They abut each other. So as the voltage changes due to the action potential, you have channels inside the sarcoplasmic reticulum and the T tubule that are voltage sensitive calcium channels. And so as the voltage changes, this opens these voltage sensitive calcium channels, and calcium is then released into the cytoplasm. So it goes out into the cytoplasm of the muscle cell. So this is the actual coupling. You've taken that electrical event, you have that action potential coming down the axon. We turned it into the chemical event where we released acetylcholine into the synapse. That then bound to the sarcolemma of the muscle cell, generating another electrical event, another action potential. That action potential then traveled down the sarcolemma, and now we've released calcium, and now we're going to be able to start those steps of actually contracting the sarcomere and then the muscle fiber. So the next step is you have the release of calcium. And if you remember, you have the thin and thick filaments of the sarcomere. So that thin is the actin, and the thick is the myosin. Let me spell that out, thin and thick. So the actin is this double beaded, like a pearl necklace, these little blue things. And associated with that, you have troponin and tropomyosin. Troponin has a calcium binding site. So the first step is calcium is going to bind troponin. Well, that causes a shape change, which allows tropomyosin to roll away from these myosin binding sites on the actin. So calcium comes in, binds the troponin, which causes the shape change, and then tropomyosin then rolls away, and you get the myosin binding sites exposed. Well, now myosin and actin, because remember myosin has an actin binding site, so that actin and myosin can now bind together. And when you get this binding of actin and myosin, we call this a cross bridge. So this is going to be the beginning of contraction. So how important is calcium to contraction? It's very important. So when you have low levels of calcium inside the cytoplasm, your troponin won't be bound, so tropomyosin won't roll away from those active sites. Your myosin and actin don't get together, and your muscle fiber will relax. So at the end of a contraction, that's what you do. You basically remove the calcium from the cytoplasm so that the muscle can relax, because the actin and myosin can't be bound. As long as you have calcium in the cytoplasm, tropomyosin will stay rolled off of the actin because troponin has been bound by calcium. So that's going to occur as long as the nervous system is sending a stimulus down the axon to the neuromuscular junction, and acetylcholine is being released, and you're getting that action potential on the sarcolemma. As long as that's happening, you're going to continue to keep calcium in that cytoplasm. So to relax a muscle, you have to stop nervous stimulation, stop that acetylcholine from binding the receptor, quit generating an action potential on the muscle side so that the calcium will no longer be released into the cytoplasm. I'm going to let you watch a short video about excitation contraction coupling. Then I will see you on the other side of the video, and we'll continue with cross-bridge cycling and relaxation.
Typically, a single motor neuron arising in the brain or spinal cord conducts action potentials that travel to hundreds of skeletal muscle fibers within a muscle. The sequence of events that converts action potentials in a muscle fiber to a contraction is known as excitation-contraction coupling. If we look at a single muscle fiber, we see that an action potential travels across the entire sarcolemma and is rapidly conducted into the interior of the muscle fiber by structures called transverse tubules. Transverse, or T-tubules, are regularly spaced enfoldings of the sarcolemma that branch extensively throughout the muscle fiber. At numerous junctions, the T-tubules make contact with the calcium-storing membranous network known as a sarcoplasmic reticulum, or SR. Where it abuts the T-tubule, the SR forms sac-like bulges called terminal cisternae. One portion of a T-tubule plus two adjacent terminal cisternae is known as a triad. The membranes of the T-tubule and terminal cisternae are linked by a series of proteins that control calcium release. As an action potential travels down the T-tubule, it causes a voltage-sensitive protein to change shape. This shape change opens a calcium release channel in the SR, allowing calcium ions to flood the sarcoplasm. This rapid influx of calcium triggers a contraction of the skeletal muscle fiber. Thus, calcium ions are responsible for the coupling of excitation to the contraction of skeletal muscle fibers.
Okay, so we're going to pick up with the cross-bridge cycle. So our actin and myosin are bound, and the actin and myosin, there's actually movement. The actin and myosin are going to slide against each other, which will shorten the sarcomere. And this is going to occur as long as you've got calcium in the cytoplasm, and also ATP. What is ATP? ATP is the energy source that we use for muscle contraction. And so you have to have energy. This is where the ATP comes in to actually cause the energy or use the energy for muscle contraction. So there's 4 steps. We have cross-bridge formation, the power stroke, cross-bridge detachment, and the cocking of the myosin heads. So I've written this out for you because I know some people kind of like to have notes written out for them, but I'm going to describe it in picture form. So these 4 steps, the first step is the cross-bridge formation. So remember, calcium has bound the troponin, the tropomyosin has moved away, your actin and myosin binding sites are available, so you get this binding of actin and myosin. Also remember that myosin has a binding site for ATP, and so this is where ATP will bind. Well, you're probably looking at this and saying that says ADP, not ATP. So let me give you a little backstory on that if you've forgotten from your Unit 1 chemistry. Do you remember what ATP stands for? It's adenosine triphosphate. And so ATP, I'm just going to draw kind of what it looks like, is an adenosine. Okay, you don't need to know the chemical structure. And then there's a phosphate, and a phosphate, and a phosphate. So adenosine triphosphate. Well, these bonds, remember from chemistry, energy is stored in those bonds. And so when you break a bond or make a bond, you're changing energy storage. So breaking a bond will release energy, and forming a bond will store energy. So these are the bonds that we're going to break in order to release energy. So what we have here is ATP is already bound to the myosin, and it's broken down. It's had the first bond has been broken into ADP and inorganic phosphate. Okay, so this is our starting place. The cross-bridge formation with myosin and actin have been bound, and the myosin has been primed or ready to go by having ATP bind and then broken down to ADP plus inorganic phosphate. So this first bond has been broken. Now the second step is called the power stroke. And so this is the actual where the myosin literally cocks and moves the actin. And this is called the power stroke. And remember, I told you if you break a bond, you release energy. So this bond that's being broken, you're basically breaking the bond that connected the ADP and then organic phosphate to the myosin. Well, breaking that bond cocks or allows the myosin to move. So kind of think about it like a rubber band. So you would say the ATP would be me pulling back the rubber band, and then if I release the ATP, released it, the rubber band would shoot across the room. So I use my ATP to pull it back, I release the ATP, and the rubber band shoots. Same kind of concept going on with the actin and myosin molecule. You've released this bond and the myosin moves. Now, actin and myosin are going to stay bound together until something comes in and causes them to break apart. So the next step is called cross-bridge detachment, and that requires ATP to come in. So when ATP comes in and binds the myosin, that breaks the bond between actin and myosin. So you have cross-bridge detachment. So this is where you need that ATP to come in and break that bond so that you can reset it, cock the myosin, and go again. So we've got ATP coming in and breaking the bond between actin and myosin. You hydrolyze or break with water, break a bond of ATP, turn it into ADP plus PI, inorganic phosphate. That cocks the head. So you've used the energy to cock the myosin head so that now it's ready to bind actin again and then move forward. So cocking of the myosin head. So the cross-bridge cycle is really a cycle. It's cross-bridge formation, the power stroke, cross-bridge detachment, and cocking of the myosin head. And this continues in a cycle as long as calcium is in the cytoplasm to bind that troponin. So this cross-bridge cycle, as long as those myosin binding sites on actin are exposed, these cross-bridges will cycle. With each cycle, the thin filaments are pulled inward and the sarcomere shortens. So you can think of the myosin like a ratchet pulling the actin closer and closer together, shortening the sarcomere. This is called the sliding filament theory. So as each sarcomere shortens, remember, a muscle fiber is made up of a lot of sarcomeres. So as these sarcomeres shorten, the entire muscle fiber shortens. And if a lot of muscle fibers shorten, the muscle shortens, and you get contraction. These cross-bridge cycles will continue as long as you have calcium in the membrane. It will end once calcium is transported back into the sarcoplasmic reticulum. Now, you get that calcium out of the cytoplasm and into the sarcoplasmic reticulum using active transport. What is active transport? That uses ATP. So again, let's review back from Unit 1. Active versus passive transport. If you have passive transport, you have a channel where an ion can flow from its high concentration to low concentration. You just open the channel and things flow down their concentration gradient passively. With active transport, you have a channel, but you need energy, or ATP, to pump that ion against its concentration gradient. So it's like a salmon swimming upstream. You got to swim against the current. So it requires energy to push that calcium back into the sarcoplasmic reticulum. Okay? By removing that calcium, your troponin will return to its original shape, and tropomyosin will cover those myosin binding sites, and actin and myosin can't bind anymore. So to completely relax the muscle, you have to stop the nerve impulse. So we've contracted the muscle, so our nervous system's not sending any more signal down. No more action potentials coming to the axon terminal. No more acetylcholine is being released, and the acetylcholine that's in the synaptic cleft is being removed by an enzyme called acetylcholinesterase. We also need to remove the calcium from the sarcoplasmic reticulum, which allows actin and myosin to no longer be able to bind. So all four of these things have to occur in order for the muscle to relax. Now I've got a short video that shows you this cross-bridge cycling, so watch this short video and I'll see you on the other side.
The contraction of a skeletal muscle generates the force necessary to move the skeleton. A contraction is triggered by a series of molecular events known as the cross-bridge cycle. In a skeletal muscle fiber, the functional unit of contraction is called the sarcomere. A sarcomere shortens when myosin heads in thick myofilaments form cross bridges with actin molecules in thin myofilaments. The formation of a cross bridge is initiated when calcium ions, released from the sarcoplasmic reticulum, bind to troponin. This binding causes troponin to change shape. Tropomyosin moves away from the myosin-binding sites on actin, allowing the myosin head to bind actin and form a cross bridge. Also note that the myosin head must be activated before a cross-bridge cycle can begin. This occurs when ATP binds to the myosin head and is hydrolyzed to ADP and inorganic phosphate. The energy liberated from the hydrolysis of ATP activates the myosin head, forcing it into the cocked position. A cross-bridge cycle may be divided into 4 steps. Step 1. Cross-bridge formation. The activated myosin head binds to actin, forming a cross bridge. Inorganic phosphate is released, and the bond between myosin and actin becomes stronger. Step 2. The power stroke. ADP is released and the activated myosin head pivots, sliding the thin myofilament toward the center of the sarcomere. Step 3. Cross-bridge detachment. When another ATP binds to the myosin head, the link between the myosin head and actin weakens, and the myosin head detaches. Step 4. Reactivation of the myosin head. ATP is hydrolyzed to ADP and inorganic phosphate. The energy released during hydrolysis reactivates the myosin head, returning it to the cocked position. As long as the binding sites on actin remain exposed, the cross-bridge cycle will repeat. And as the cycle repeats, the thin myofilaments are pulled toward each other and the sarcomere shortens. This shortening causes the whole muscle to contract. Cross-bridge cycling ends when calcium ions are actively transported back into the sarcoplasmic reticulum. Troponin returns to its original shape, allowing tropomyosin to glide over and cover the myosin-binding site on actin.
Okay, so finally, we like to talk a little bit, as we learn about anatomy and physiology, some practical application. So one of the things I wanted to talk to you today was about rigor mortis, and this is a term that literally means the stiffness of death. Mort means death, rigor stiffness of death. And this occurs about 3 hours, and it maximizes about 12 hours after death. And this is because when you're dead, you are no longer breathing. And so the oxygen levels in your body will start to decline because you're not breathing and getting in any more oxygen. Well, if you don't have oxygen, then you can't do something called cellular respiration, which you learned about in Unit 1. Without cellular respiration, you can't make ATP. So without ATP, actin and myosin will stay bound together. Remember, when actin and myosin are bound together, you need an ATP to break that bond. Also, without ATP, there's nothing to pump calcium back into the sarcoplasmic reticulum. So you have calcium out in the cytoplasm binding troponin, causing tropomyosin to turn. Actin and myosin are bound, and there's no ATP to break that bond, so the muscles all contract. So rigor mortis is that stiffening or contracting of the muscles after death. And it lasts about 3 days, about 72 hours, and then the muscles will start to relax again. And that's because, not because you're breathing and making ATP, but because you're basically starting to rot. So the muscles will start to break down. And actin and myosin are proteins, and just like any kind of meat or protein, it will start to decay. So at 72 hours, those proteins decay and they just break apart, and you start to get some relaxation. A lot of times we don't see that because the bodies that we see in rigor mortis, we usually bury our dead before 72 hours. So a lot of times the bodies we see are still in rigor mortis because we tend to bury our dead within 3 days. Okay, so that's all I'm going to talk about today, and I'll see you in another video. Thank you for your attention.